Thank you for that introduction, Stephen, and thank you for inviting me to talk today about our leukosis project. I'm happy to provide a little update on what we've been doing and what we will continue to keep doing for several more years. So what I have planned to talk about today, first I'll just do a little bit of an introduction, talk about what bovine leukemia virus is. I'll hopefully tell you what questions we are answering and how we're trying to do that. I'll give you a little bit of insight on what we've found so far, and I'll tell you what we plan to do next. So bovine leukemia virus, as you can guess, is a virus, and it is the virus that causes enzootic bovine leukosis in cattle. It's found in beef and dairy cattle, but we find it more often in dairy cattle. And something that's really important to note is that infection with BLV is permanent. So once a cow tests positive for BLV, she has it for the rest of her life. So there is no curing BLV, there's only preventing BLV. And we know that cows that are infected with BLV, they are less productive overall. They're more susceptible to infection with other diseases because BLV causes their immune system to be compromised. BLV positive cattle are less fertile, so they are less likely to get pregnant, and they are more likely to be culled before it's optimal. So the dairy farmers of Canada, they estimated that leukosis on average costs about $12,000 to $19,000 per year, assuming that there is an average of 100 milking cows. So it's kind of important. And the next important question is how prevalent is BLV in Canada? And the answer is that it's very prevalent. In 2017, researchers estimated that the prevalence of BLV is about 89% at the herd level, which means that in almost 90% of herds in the country, at least one cow is BLV positive. But in reality, on most of these BLV positive herds, about 20 to 30% of the animals are infected with the virus. BLV, we know, is spread through the transfer of infected blood cells, so infected B lymphocytes, and this transfer of blood products can happen through multi-use needles, which often happen for vaccinations or ovulation synchronization protocols. We think that multi-use rectal sleeves that are commonly used for like AI and pregnancy diagnosis might be a source of infection. We say might be because in the literature there's actually not a lot of evidence for it, but anecdotally talking to producers, we know that this is a really common control method that they use, and they found that it works to reduce the prevalence on their farms. We also know that I'm reading the wrong bullet. Improper disinfecting of equipment, like hoof trimming equipment and dehorning equipment might be a possible vector for infection or a method of infection, and biting flies. So currently, based on what I just said, we don't really know when new BLV infections are most likely to occur, and we don't know how they are occurring. And it's unclear, therefore, what the best way to prevent new BLV infections from happening is. So that makes sense then that our questions are how are new BLV infections acquired, and what is the best way to prevent new infections from occurring. So that is part of my job during my PhD, is to try and figure this out. So a large team has started a long-term longitudinal study to look at the transmission and the control of BLV on 10 commercial dairy operations. And in this study, we want to observe new BLV infections. We want to examine how the viral load of these cows changes over time. And we want to explore differences in transmission of BLV between adult cows and heifer calves. So we've defined this as heifer calves that are younger than 6 months of age. So what does this look like on farm? For heifers and cows older than 6 months of age, they're enrolled, so they have a blood sample taken. And they have a blood sample taken every 12 weeks until the end of the sampling period, which is about 18 months. For calves less than 6 months of age, they're also enrolled at day 0, and they have a blood sample taken. But for the first 6 months of their life, they're sampled more frequently. They're sampled every 3 weeks. And this is because we think that the younger calves, because they're immunocompromised, or they're immunonaive, they don't really have like a properly functioning immune system, and they're under a lot of stress, we thought that maybe transmission events might happen more often. And then once these calves become 6 months old, they kind of enter that every 12 weeks protocol until they're about 24 months. And then just to reiterate that this is a lot of blood samples being taken. And at each blood sample, we take 2 tubes. We take 2 vials of blood from the animal. The first is a red top tube that we use to look at serum. And this serum is sent from all animals to the animal health lab immediately after collection. And that is where they perform serum ELISA, where they look for antibodies against BLV. And the result of this test is that it tells us yes or no if the cow has the disease. The second tube of blood is a purple top tube, and it's used for a test called PCR, and it determines the viral load of an infected animal. So only animals that were determined positive on the first test have this test done, and it tells us like a numerical value of how much virus is inside the cow. And this is a test that we do like just for research purposes. And this slide I wanted to show just how much blood we're taking. This was my most proud moment. We collected over 600 blood samples. 600 cows were sampled, so over 1,200 blood samples were taken that day. But overall, we still managed to have a pretty good time. So now the exciting part to talk about the results. So this is a little overview of the 10 herds that are involved in the study. As you can see, they range in size from about 150 milking cows all the way up to 500. The majority of them are on DHI, which is how we kind of recruited them. We knew from a previous study the result of their bulk tank milk pickups, and then we were able to do like a milk test. And then after we knew that they wanted to participate and that they had leukosis on their farm, we went out and we did like a whole herd blood sampling where we found that the results were pretty similar. And we've actually had the opportunity to go through this whole cycle of 12 weeks at least once on each farm. And we found that in these 12 to 16 weeks between whole herd samplings, some farms have had 0 new infections, and other farms have had up to 31 new infections in that time. It kind of seems to correlate to how prevalent BLV is on the farm, but not exactly. And so part of the work that we still have left to do over these next few years is to see why some farms are having so many new infections and some are having 0. And then to talk a little bit about the results of what we've seen in the calf data so far. So this is data from just one farm. It's actually farm B that I just showed you. These are the results of calves that we've sampled from 0 to 6 months of age. And as you can see, there's kind of 4 things that can happen with them. They either are always test negative, always test positive, they start test positive and they become test negative, or very rarely they start test negative and become positive. I think I might have said that backwards. They start positive and become negative is more common. And no, I keep mixing that up. Sorry, they start positive and become negative. You guys can just read it. This is just one. This weird one is actually just one calf, and we think what might have happened is that we sampled her before she got colostrum, maybe. So we sampled her on day 0 when she received no antibodies from the colostrum. Then she was sampled again and became a very high responder. And then over time, as you could see, she went toward negative. So 44% of the calves that we had in this cohort were always positive. About a quarter were always negative. About 28% went from positive to negative, and just that one calf went from negative to positive. So we're still at the very early stages of this, so a lot of the questions that I mentioned at the beginning of the presentation we still have. We'd like to know when animals are most likely to test positive, and we want to know what management practices are most associated with new infections. That will help us answer the question of what is the best way to prevent new infections from occurring. And something of interest is maybe how an infected animal's proviral load changes over time. Because we know that culling high proviral load animals is an effective management strategy. So knowing how this changes might help producers make culling decisions. So our next steps are basically just to continue with the project and see what happens. So this is a huge project that involves a ton of work. So I'd really like to thank everyone that's been involved, especially my advisor, Stephen, Emma Morrison, Kylie, Melody Knott, and all the undergrads that have helped us this summer, and my advisory committee, and all the funders, and all the producers and their cows. Thank you.